As we all know, the treatment of relapsed or refractory multiple myeloma still remains a big challenge for now. So in this session meeting, are there any notable advances about relapsed refractory multiple myeloma? Would you please talk about this from the aspect of different types of agents?
Absolutely. No, I
Oh,
think there are a lot of new data coming out at ASH related to how we manage relapse and refractory disease. As there are a lot of new drugs that have come available for patients over the past 10 to 15 years, particularly the proteasome inhibitors, the immunomodulatory drugs, as well as the more recently the monoclonal antibodies, if a few years ago. Now, clearly, patients are getting these drugs, and the myeloma is starting to break through the proteasome inhibitors, the monoclonal antibodies, and these IMiDs. And as the disease becomes refractory to these agents, we have to start developing new drugs or new modalities that work through entirely different mechanisms of action. So I would think broadly classify them as three different groups of drugs. So one of them clearly represent the improvement over the existing classes of drugs. So you clearly have oral proteasome inhibitors, new proteasome inhibitors that are coming through. We have new immunomodulatory drugs, especially what now being called as the cell mods. We saw some follow-up data from iberdomide or CC220. That is a new cell mode that appears to be quite effective even in patients who are refractory to lenalidomide and pomalidomide. And there are some basic science work related to the mechanism of action of iberdomide as well. And then there are new anti-CD38 monoclonal antibodies that are being developed, including drugs like the MOR202 and TAC079. So all these are, again, anti-CD38 targeting, but monoclonal antibodies that might represent an improvement over the current generation of anti-CD38 monoclonal antibodies. But I think the most exciting is the third, are the new small molecules and the immunotherapies. So amongst the new molecules, new classes of drugs, we certainly have, some data with venetoclax that seems to be effective, a BCL2 inhibitor that's effective in the translocation 11-14 patients. We certainly have more data pertaining to the tyrosine, especially the targeted agents like the MEK inhibitor, as well as the, knowing that there's almost half of the patients with myeloma have mutations involving KRAS or NRAS, this class of drug might have a significant role to play in this disease as well. And then there's some data, again, kind of going back to that first class of improved existing classes would be melflufen, that is a new alkylating agent that also has some data from clinical trials being present at ASH. But I think as I said, the major improvement I think is going to come from the immunotherapeutic approaches. And again, there are two major groups. One is the CAR T cells, or the chimeric antigen receptor T cell therapy, and then the bispecific molecules. And we have exciting data from both classes of drugs. So we have data being present at ASH, which is longer-term follow-up of the BB2121. The KARMA trial results clearly show that there's durability of response. Majority of the patients treated with the BB2121, nearly 70 to 80% of these patients actually respond to therapy. And the median duration of response appears to be roughly about a little over a year, and the median PFS of about 9 to 10 months. So I think clearly we were hoping that maybe the responses might last even longer, but even this durability of response is quite something, considering that these patients had median of 5 to 6 prior lines of therapy and are refractory to the current classes of drugs. So certainly impressive results, and the BB2121 seems to be the closest in terms of FDA approval as well. Now then we saw data from the cilta-cel, that is the original Legend molecule now being developed by Janssen. And the results of that, again, over 100 patients, almost 100% overall response rate, and very deep responses. And again, we don't have a median progression-free survival, but based on what we are seeing so far, it clearly appears that the median PFS would probably exceed a year and a half in a heavily pretreated group of patients with median of 6 prior lines of therapy. So that is also very promising responses. Now, we also saw data from some newer CAR-Ts that are being developed, including the CT053 that is being developed by Carsten. Then there's the PBNC 103 that is a new CAR-T cell that Dr. Caitlin presented for their data from the UC San Francisco that also seemed to be quite interesting because it utilizes a different mechanism than lentivirus and uses a transposon-based approach to creating the CAR T cells. And that certainly could alleviate some of the problems associated with lentivirus, or at least the potential for problems which require these lentiviral vector-based CAR T cell patients to be followed up to 15 years. So hopefully these new platforms for delivering that would definitely make the longer-term follow-up easier and maybe make these things safer. But another exciting aspect of the CAR T cell was the introduction of the allogeneic CAR T cell. So we saw the results with ALLO-715, that is a allogeneic off-the-shelf CAR T cell that again, based on the initial group of patients that were treated, appears to be effective. We've seen some responses. Some of the responses have been lasting. We haven't seen any significant toxicity, particularly graft versus host disease, which was something we were worried about. Now, one thing that is common amongst all these CAR T cell is nearly all patients get some degree of cytokine release syndrome, but that can be very easily managed. Severe CRS of grade 3 and 4 appears to be relatively rare. Now, the neurotoxicity is something that we certainly worry about, and most of these agents appears to have very low rates of neurotoxicity, usually grade 1 and 2, though we did see some delayed neurotoxicity with cilta-cel, which certainly needs further study to better understand that. Now, the other group of immunotherapy are the bispecifics. Now, obviously, we saw some updated data from the Amgen for 20 molecules on the Amgen 701. So clearly, that was one of the earliest bispecifics that were tested in myeloma, and the results still appears to be quite promising. We saw some newer BCMA targeted bispecifics, especially the teclistamab from Janssen. Again, over 100 patients, very high response rate, 70 to 80%, manageable cytokine release syndrome toxicity, nothing specific for neurotoxicity. There's obviously a signal for increased risk of infection, which we obviously need to get a better understanding of. We also saw a bispecific against BCMA from TinyBio, the TNB038B, that also appears to be quite effective with not as much of a toxicity signal based on the number of patients treated so far. We also saw newer targets being targeted with these bispecifics, especially the talquetamab, which is targeting the GPRC5D. And then there's also the FCHR5 CD3 bispecific from Genentech Roche that also, again, showing some early results that appears to be quite promising. And then finally, there's a newer antibody drug conjugate, the MEDI2228, which is again a BCMA targeted antibody drug conjugate that appears to be quite, appears to have good activity, however, also has some eye toxicity that is being worked through as well.
Nowadays, the BCMA is the key target for CAR T therapy towards myeloma. And in this ASH meeting, you presented one study evaluating the efficacy and the safety of CT053 in heavily treated multiple myeloma patients. And the efficacy is very impressive. So would you please talk about this study? And is there any advantage over BB2121?
Certainly. So the study that we're doing is a phase 1B with a planned phase 2 expansion of CT053, which is the BCMA targeted CAR T cell from Carsten Therapeutics. Now, there were two presentations at ASH. Dr. Hu also presented some longer-term data with the LumiCAR-1 study that is being conducted in China, and the LumiCAR-2 is the one that is being conducted in the US. LumiCAR-1 had 24 patients. The LumiCAR-2 that I presented had 20 patients. And both the trials showed that the overall response rate in the range of 80 to over 90%. In the LumiCAR-2, again, 17 of the 18 response evaluable patients actually had a response. And impressively enough, 10 of the 17 patients actually had a very good partial response or better. And nearly 14 of the 18 patients, their bone marrow had already become MRD negative, despite having serum levels of measurable M protein. Now, so efficacy is clearly good so far. The phase 2 trial is now going to start enrolling with the recommended phase 2 dose of 2.5 to 250 to 300 million CAR T cells. Now, in terms of toxicity, we again saw mostly grade 1 or 2 cytokine release syndrome. There was one grade 3 neurotoxicity, but overall, this was all very easily managed. Most of the toxicity appeared to be cytopenias, and that is related to the lymphodepletion. We saw a couple of patients with some delayed thrombocytopenia and neutropenia, which we believe might be related to the CAR T cell itself. And this was a very heavily pretreated group of patients. Almost 3/4 of the patients were triple refractory, over half of the patients were penta-refractory, and nearly half of the patients had high-risk cytogenetics, and a quarter of them had extramedullary disease. With the treatment we saw by 4 weeks, all patients had clearance of their bone marrow. By 4 weeks, most of the light chains had normalized in patients with the measurable light chain levels. And patients with an M spike that is more than a gram, you could see profound reductions in the M spike level in the setting of CT-053 treatment. And the lymphodepletion was pretty standard. Fludarabine cyclophosphamide given as an outpatient. The patients were hospitalized for about a week after the infusion of CAR T cell, primarily for observation. They really didn't have much of a problem except for the grade 1 or 2 cytokine release syndrome. So overall, the safety profile looks interesting, and the activity so far is quite encouraging, so that the trial is going to move ahead with the phase 2 trial.
Now, in the era before daratumumab, the 4-drug combination in newly diagnosed myeloma patients is not recommended due to the toxicities. But in recent years, daratumumab-based 4-drug combination has provided deep and durable responses. So do you think we are ready to step into the era of 4-drug combination in first-line setting? If so, what kind of patients would you prefer to use the 4-drug combination? Thank you.
Absolutely. So I think, the data for the quadruplets are still emerging. So we have data from the Griffin trial looking at daratumumab with bortezomib, thalidomide. Dexamethasone clearly shows progression-free survival advantage for the 4 drugs compared to the 3 drugs. We don't have any overall survival data yet. The important question that comes up is, can the 4th drug be used later on instead of using all 4 drugs at the same time? And similarly, the Griffin trial with the daratumumab VRD, again, clearly deep responses before going to an autologous stem cell transplant. We also have data from the Alcyone trial where the daratumumab was added to VMP in the non-transplant setting and showed an improved overall survival as well. So I think there's clearly a role for introducing daratumumab in the upfront setting, whether it be part of a triplet or a quadruplet. I think we still need to wait for the longer-term data with the daratumumab VRD before we say that a 4-drug regimen should be the standard injection therapy for these patients. Now, there are clinical trials ongoing that's looking at adding for the 4th drug based on the depth of response. Clearly, one of the piece of data we need to have is whether we can use these 4 drugs together, or how can we compare that to using maybe 2 triplets back to back containing the similar drugs. So those are the things that we really need to be looking forward to right now.
In this ASH meeting, I noticed one interesting abstract presented by Professor Sammer from Dana-Farber Cancer Institute, and they concluded that high-dose methylene significantly increases the mutational burden in multiple myeloma cells at relapse. So in the era of so many novel agents, which can put patients into deeper remissions, do you think it's time for us to reappraise the role of high-dose methylene?
I think it's a question that's continuously going to be asked as we have newer therapies. I mean, at this point, myeloma still remains incurable. Even with the more effective therapies like CAR T cells, we are not able to keep the disease under control for long. So I see no reason to throw away a very effective therapy like transplant that can keep the disease under control for 2 to 3 years, or just by itself, and can produce deep responses and even deeper responses in the context of these more effective therapies. So I still think about it as one element of the therapy that needs to be used in conjunction with the other treatments that we have. The presentation looking at the mutational profile of the myeloma cells at the time of relapse is certainly interesting. The caveats are it's based on a very small group of patients, a small number of patients. We don't know what the implications are for those in terms of the clinical outcomes. So far, we know like the IFM 2009 trial with a very long-term follow-up, looking at transplant early versus delayed transplant, does not show any difference in the outcomes of these patients in terms of overall survival. There is also longer-term follow-up data from the EMN02 trial, which compared transplant to VMP consolidation that is present at ASH this year, did not show, shows that there is an improved overall survival for the transplant. So I don't think we are ready to throw the transplant away at this point, but we certainly need to figure out how best to use the transplant. Do we transplant patients when they get to a minimal residual disease with this 4-drug or 3-drug induction therapy? And also the important question is like the findings that were presented with the mutation load in the relapsing myeloma cells. What if we did the transplant at a time when the myeloma is in a minimal residual disease negativity stage? Will that decrease the risk of these mutational changes happening? If there's very few myeloma cells left behind at the time of high-dose melphalan, maybe that is an immaterial thing.
The last question is about the basic research. We know the pathogenesis of multiple myeloma is extremely complicated. And in recent years, the tumor microenvironment is the key element in the process of development and relapse. So would you briefly talk about tumor microenvironment in multiple myeloma? Thank you.
Yeah, absolutely. I think there's, it's been known for a long time that the tumor microenvironment plays a role in this disease, but the initial focus had been in the context of drug resistance, that the microenvironment actually protects the myeloma cells from the various drugs we use. Over the years, we have understood that it has a major role to play, not only in terms of development of the disease, the progression from precursor phases to active myeloma, as well as the development of relapsed refractory disease. So the major component of the tumor microenvironment that seems to be playing a role is the immune system. So we know that the immune system likely play a major role in terms of the transformation from Muggers to smoldering myeloma to active myeloma. There's several presentations at ASH this year that is looking at the immune microenvironment and its role during this transformation process. And the second piece is understanding the role of immune system and how it is affected by the disease as well as the treatments that give for myeloma. This has become of great interest because now we have all these immunotherapies, some of which include collecting autologous T cells for the manufacturing of CAR T. So there are important questions being asked. What time point are the T cells the most competent? What kind of influence do the myeloma cells have on these immune cells? And how can we actually modulate these immune cells in order to enhance the anti-tumor activity, whether it be CAR T cells, bispecifics, vaccinations, NK CAR Ts, and so forth. So that's one aspect. The second aspect of the tumor microenvironment is clearly the bone disease. And obviously there have been a lot of focus on that in the past, and we have bisphosphonates and the anti-RAN ligand inhibitors, both of which appear to be effective in reducing the bone disease. The impact of these measures on the disease itself is not very well outlined, though we've seen some PFS advantage, both for the bisphosphonates as well as for denosumab in the phase 3 trials. The third aspect is the Marrows trauma. We know that clearly seems to help the myeloma cells escape therapy. We know that there is increased angiogenesis in the tumor microenvironment or the bone marrow as the tumors evolve. We also know that the myeloma cells become more extramedullary as the disease becomes progressive. with soft tissue plasmacytomas, as well as involvement of other organs and the CNS. So the mechanisms by which the myeloma cells escape the tumor microenvironment is equally important as we follow these patients long term. And we need to continue to understand the biology better.